start off by bringing up Dr. Andrew Perry from Monash University. Andrew was in the lab that Lev mentioned before that was recently featured on ABC TV. Andrew works on structural biology projects, and he is also the advisor for the Monash iGem team. iGem stands for the International Genetically Engineered Machine Contest. So, welcome, Andrew.
Would you prefer to use the lapel? It's worse.
I might actually. Okay, so yeah, Jeremy invited me along here to speak about open source biotech and synthetic biology. And so I'm hoping over the next, I think I've been given about 8 minutes, I might take a few more, to bring you into the mindset of how synthetic biologists are working today to build things out of biological parts. So you're probably all familiar with open source software. It's pretty hard to use a computer today without using it directly or indirectly. So the idea of open source hasn't really stopped there, but is expanding into other fields, and one is biotech. So this presents new challenges relative to open source software. A few examples, for instance, collaboration. There's no source code repository for actual DNA, where you can have actual DNA spat out of a computer yet. But there's no cheap desktop DNA synthesizer. We can store the sequence, but not the physical product. It really requires specialized equipment and materials, things not like computers that have somewhat become commoditized. People have to work within existing biosafety frameworks, and also there is an existing forest of patents. Because there's real advantages over closed models as well. The democratization of biotech, allowing more people to gain access to the benefits for producing things themselves. By making the information open, the technical and financial barriers to entry are lowered. We can harness the creativity of the crowd, or these people out there on the internet, and this will enable researchers in the developing world. So one of the communities that's really embraced the open source model most heavily is this emerging field of synthetic biology. So this is really the application of engineering principles to biotechnology, using things like standards, abstraction, modularity, measurement, and modelling. And really people working in this area are seeking to formalise production of devices, make things more predictable so that things can be treated as an engineering discipline rather than more ad hoc approaches that have been taken in the past. So I thought I'd throw this in here, since there's often a lot of discussion about Moore's law. Similar things have been applied to DNA sequencing and DNA synthesis, where you can see that over time these are progressively decreasing, and I think the real enabling technology for synthetic biology has been gene synthesis. We're now down, I can order today something at about 35 cents a base pair. So one of the more interesting aspects, I guess, that's come out of the synthetic biology community is this iGem competition, the Internationally Genetically Engineered Machine Competition. So this is a competition where undergrads and some high school students are challenged to build some sort of biological system or device for real from basic parts, and these operate in living cells. And so you can see here, these two guys are from the Cambridge team in the UK, and they've taken E. coli, mostly harmless, and they've put some luminescent genes from I'm not sure whether this is the firefly or the luminescent bacteria, but it glows and it works. All of the parts that they design go back into this registry called the Registry of Standard Biological Parts, And each year, a large selection of these gets sent back out to teams that they can use to build new things. The other angle I think is really important in open source biotech is this DIY bio movement. And for me, this is really the soul of open source biotech. These are groups of people who've come together under this DIY bio banner, sensibly replacing the biohacker term that they were using earlier, to a mixture of amateurs, often paired with a molecular biologist, and citizen scientists trying to do synthetic biology. Very much a DIY effort. Is the lab that's been set up in New York City called GenSpace. It has all of the biosafety certification that they need for that area, and they're doing things. I really see this akin to the early days of the information technology revolution, where guys in their garage could actually put things together and have an impact. So a few examples of the hardware that they've been coming up with. A guy called Carful Garvey in Ireland has designed this thing called the Dremel-Fuge, which he's printed this with his MakerBot, his 3D printer. And it's a simple thing. You attach it to a drill or a Dremel and it can spin up to 50,000 G. This is really useful for a lot of molecular biology protocols for pelleting cells down, separating cells, and some types of DNA purification. So he actually has put the models online, and you can buy this, get printed some other tougher material from Shapeways. And it's all under Creative Commons, so other people have forked the design and made variations on this Dremel-Fuge. Another really nice example that's going on in the hardware space is OpenPCR. Polymerase chain reaction is a standard way to copy DNA. It's also used for diagnostics and other things. And the cheapest sort of lab instruments cost about $5,000. So Tito Jankowski and John Perifetto, they wanted something cheap and open, and so they made it, they designed it, got being crowdfunded on Kickstarter, and you can get all the details of how to build this, or you can buy it from them for $512 unassembled. So this uses a lot of commodity hardware, Arduino-based controllers, and open source software to drive it. But enough about the physical hardware. How about the wetware, the real biological parts and devices? So this concept of parts has emerged in synthetic biology, and it really is analogous to Lego. You have some unit which can connect to other parts, and you can build up bigger structures and functional things. So modular, reusable sequences of DNA, usually they code for useful proteins or RNA once they're functioning in the cell. These can be things like inputs, sensors for heavy metals, salt, light, temperature, biosynthesis, production of beta-carotene. There's an iGem group in Taiwan that's been looking at producing beta-carotene in lactobacillus, so you can have beta-carotene yogurt, hydroxyurea-plastic. There's also parts for doing computation. They're not quite there yet to make computers like we know and use today, but people are starting. There's output reporters like fluorescent luminescent proteins and things that make strange and pleasant odors. I should note that all of this work in iGem teams is usually going on an E. coli, occasionally yeast. Once or twice people have looked at plants, and maybe once mouse stem cells, but the majority of groups are focused on bacterial systems. So a few of these biological parts are built de novo. You don't just sit down and write out the sequence and know what it's going to do. They're all stolen from nature. So, the MIT team that made wintergreen and banana smelling E. coli took genes from petunia and yeast. Anytime you see the things like the GFP rabbit or the glowing bacteria under UV light, this is a fluorescent protein from coral or jellyfish, and these have been turned from just any old gene sequence into a hybrid sequence that can be used as a part. Luciferase from firefly, and closer to my heart, the Monash iGem project, where they've taken ACC synthetase and oxidase from plants, which may produce ethylene. So by biobricking these parts, they have a certain nucleotide prefix and suffix added to them, and this makes them suitable for standard assembly, which is a repeatable operation. And all of these parts are documented in the parts registry, so you can get the sequence. And for instance, reporters, you can see we have RFP, GFP, a cyan fluorescent protein. The list goes on, many variations. And the best parts are also somewhat characterized. So in this case, on the part page, the MIT 2016 got people to smell the cultures of the E. coli to actually show, yes, number of respondents, yes, this does smell like wintergreen, yes, this does smell like banana. They also use more advanced methods. So in order to connect parts together in a predictable way and share them, we need standards. Much like there's only a few standards for bolts and screws so that they fit together. And the most prevalent standard is called BBF RFC 10, where the RFC request for comments style communication has been adopted similar to internet protocols. And using these standards, parts can be connected together and built up. So it's the other important concept to consider in this respect is abstraction. When connecting parts, we're no longer thinking about the raw sequence of the nucleotides, but instead treating them as units. Once we understand their behaviour, those units, parts, can be built up into devices, which might take an input and give an output in a defined way, and then those devices can be built up into systems. So an example of that might be if you have, this is based on a UCSF and University of Texas project, a light receiver, which is a number of parts, receives light, produces an output called pops, which is polymerases per second, and that pops can plug into lots of other things. So they plugged it into a colour converter and made a bacterial film act like a photographic film. But you could equally take that same light receiver that generates pops, plug it into the banana smell generator, and have E. coli, which I guess would smell like bananas during the day and like normal E. coli at night. So ultimately, putting parts together, we can build up more complex devices with behaviours. To finish on a more complex device, this is an Edinburgh iGem team project where they wanted to make an arsenic biosensor. And so skimming over the complexity of what's in here, essentially in low arsenic, this urease gene is on, which causes the pH in the culture to become more basic. When the arsenic is present, the arsenic sensor causes the LAC-Z gene to make lactic acid. Lactic acid raises the pH of the culture, and they were able to show that in fact this sort of worked. They could get changes in pH for very small amounts of arsenic. So at this point, I'll leave you with the question, what will you build?